My name is Morgan Cheng. I'm also an alumnus of the Picower Institute. I was one of the early members of the Picower Institute, and I spent 7 years here from 2001 and 2000 to 2008. I remember when this place was a hole in the ground, actually. Those were truly formative years for me, and I have just amazing fondness for the people that I met and interacted with, many of whom are still here. And so I'm just incredibly happy and proud to participate in this symposium. And very glad to hear from Li Hui's introductory remarks that I am a beloved alumnus. When I was at the Picower, my lab studied synapses, which are the communication junctions that neurons make with each other to, form circuits and make memories in the brain. Synapses have this remarkable property of being able to make or break, strengthen or weaken, and it's largely through this property that we're able to have a plastic nerve system. And plasticity is the theme of the session today. We have 3 speakers, and we'll get straight to those speakers. The first of which is Jerry Chen, who is assistant professor at Boston University, and he will be introduced by his former Picower Institute advisor, Ellie Nadivi.
Thank you, Morgan. It's really a pleasure for me to be here today and to be able to introduce my former student, Jerry Chen. When Jerry was in my lab, he pioneered both a molecular approach and the microscope development that allowed us to label and track individual synapses, the contacts between neurons, in the living brain. And not only was it a first for our lab, these were the first experiments of this type at all. And it really gave us kind of an unprecedented view of how synapses kind of rearrange in the living brain. And I think that this typifies Jerry's work, not just when he was in my lab, but also later in his postdoc with Fritjof Helmchen in Zurich and in his own lab at BU, where he develops a technology which kind of gives a first view of things that were previously not visible, and really allows kind of a step forward in our thinking and understanding. And I think, like with your kids, you can take credit for how well they turned out. But in the end, if you have kids or students, you know that you can give guidance and support, but their success is really their own. Jerry.
Thank you, Ellie, for the introduction, and thank you for the opportunity to come and join you guys on this 20th anniversary of the Picower Institute. It's really great to be back here. So kind of keeping in spirit of trying to look back and, at my time at MIT and the Picower, I thought I would start with this. Okay, great. All right, so I pulled this out from the archives. So this is the Nadivi lab circa 2006, right around the time that the building opened. And here's me with a little more hair. Here's Ellie, who looks exactly the same as she did back then. And so I joined Ellie's lab in 2005, and I mean, looking back at this photo, it's kind of hard to imagine what I was thinking back then and how I would think things will kind of evolve over the years, both in terms of the science in general and also the research work that we've been doing. And in general, I think one of the interests of many of us is to understand common principles of the brain, right? And at the level, for me, at the level of circuits, so what are the things that are generating our thoughts, perceptions, interactions, learning, and memory, right? But also, as I've been kind of thinking about this and working on this problem, I've started to also been interested in understanding individual differences. How is Liwei's brain different from Morgan's brain, and how is Ellie's brain different from Casey's brain, okay? And so my research program is really kind of focused on the idea of trying to understand common principles of brain function, but use that common principles to understand how there are differences between us, our individual variation. And at the same time, try to look at individual variation and try to, and from that, derive common principles. So, systems neuroscience is really about the study of circuits. And in order to study circuits, you really need 4 types of information. You need to know the activity patterns of the neurons that you're studying. You need to know how they're connected together. And you need to know their gene expression patterns that define our identity. And all of this needs to be tracked in the context of behavior. Okay? So currently, I would say the approaches that we're taking is a more horizontally integrated approach, where we're taking these different measurements in different subjects, individuals, and then you're trying to assemble an average image or a picture of what these stereotyped circuits are doing and operating. Now, an alternative approach is a more what I call a vertically integrated approach, in which that from an individual subject, you're trying to make all these different measurements of behavior, activity, connectivity, and gene expression together to look at the circuit function of that individual subject. And this might just try to seem like this idea of you're trying to image everything everywhere all at once, but I would argue that there are certain questions that you couldn't address without this more vertically integrated approach. And the idea is that in order to study individual differences, you need to treat each circuit in an individual as a unique sample. And in order to look at these individual differences, you need to be able to make all these different types of measurements across modalities. So our lab has been focused initially on trying to push forward on these more vertically integrated approaches. How did we get here? So if we go back to 2006 in Ellie's lab, if I could kind of summarize what Ellie's program has been about, it has been to develop advanced multiphoton microscopes in collaboration with Peter So here at MIT, with the intention of trying to look at structural and synaptic changes in an awake living brain, with the idea that these structural and synaptic changes that you're seeing are derived from candidate plasticity genes. Now, when I was in Ellie's lab, my focus has really been was on the structural and synaptic changes that are occurring in the primary visual cortex. And I was around kind of observing what others were doing in the lab in regards to these other areas of research, but not really directly involved. However, fast forward to 2022 in my lab, somehow, I don't know how, we ended up kind of tackling certain questions or technologies that that Ellie has been working on this whole amount of time. And so I'll be talking about 2 small stories here where we're trying to looking at neuroactivity and gene expression under the context of this more vertically integrated approach. Okay, so my lab doesn't work in a visual system. We work in a mouse whisker system, which is a great system for us looking at sensory processing, sensory motor integration under the context of behavior. And we train mice to relatively complicated sensory tasks like tactile working memory tasks. And what you notice here is that these mice are head fixed. And the reason why they're head fixed is because we're using imaging technologies to study activity patterns in the brain as the animals are performing these types of tasks. In terms of imaging, many of you in the room might be familiar with calcium imaging, right? So when neurons spike, calcium enters the cell. You can use calcium as an indirect readout of spiking activity. If you notice the dynamics of calcium with respect to spikes themselves, the voltage signals, you notice that there's a difference in time scales, right? The calcium signals are way slower than these voltage signals, okay? And this is somewhat convenient for a lot of current approaches in microscopy that can image on the order of 10 to 30 hertz, right? This is sufficient to be able to capture these calcium signals. However, for voltage, given the millisecond spike timing activity patterns that exist in the brain, and with the rise of new voltage sensors that allow us to more directly image voltage, now we're confronted with this issue of trying to increase our imaging speed to a kilohertz, right? And our lab has been working on pushing microscope technologies that increases the frame rate of imaging, but this is not a new idea. And in fact, a lot of these ideas were actually really pioneered with Ellie and Peter back in the day, in which, in order to increase the rate of imaging, rather than using a single laser beam to scan your area, you can parallelize using multiple beams, and this increases your temporal resolution. And so Grace and Allison and the lab worked together to build what we called an ultra-fast two-photon microscope, again, adopting similar principles where we're scanning fields of views in parallel using multiple beams. So with this particular microscope, we can image 400 by 400 microfield of view, image maybe on the order of 100, 200 neurons, but now we can do so on the order of a kilohertz frame rate. Other advances recently in terms of deep learning and image denoising allows us to really pull out the spikes. So when we're imaging this quickly, you get really low levels of signal here, but you can infer the underlying noises that exist in the system and in your sample to pull out the actual voltage signals that you show here, using these more recently developed voltage indicators. So from now, we're using a combination of these voltage indicators, our microscope, and this deep learning denoising algorithms. We can start to pull out spiking activity in the field of view of about 100 to 200 neurons. OK, so I talked to you about neuroactivity. Now I'll talk to you about gene expression. So we're interested in integrating measurements of neuroactivity with gene expression because the gene expression patterns of individual cells help to define the identity of these neurons in the brain. And in recent years, there's been a large effort to generate what people refer to as a cell census atlas, in which, using RNA sequencing technologies, people have been able to measure or try to identify the types of cells there are in the cortex. And the Allen Institute here has, through their RNA sequencing work, has estimated they're on the order of 300 to 400 different types of cells in a mouse cortex. And so this raises the question as what are the function of these particular types of cells? So in order to kind of tackle this, we developed a technology called CRAC, which stands for Comprehensive Readout of Activity and Cell Type Markers. And I'll kind of show this animation in terms of how it works. So here we're imaging neuroactivity in the brain, in this particular case calcium imaging, as animals are performing a task. And then once, at the end of our behavioral experiments, what we can do is we can take the brain out, we can section the brain, and then we can find the same neurons that we imaged in vivo in these fixed tissue sections. And then from there, what we can use is a technology called spatial transcriptomics, which is the ability to read out mRNA expression in situ in this tissue section, right? So we're using a technology called HCR-FISH, which gives us a very high signal to noise in terms of staining. And we're using confocal microscopes where we can then image individual mRNA molecules in the brain. Now, the power of this type of staining is that we can multiplex. So we can stain for a certain set of genes, we can strip, and then we can restain for a different set of genes. And by doing this across multiple rounds, we can build up a gene expression profile of the neurons so we can know what types of cells that we've been recording from. And so from this, then, we can essentially combine the data that we collected in terms of the neuroactivity patterns and correlate that up with the gene expression patterns. OK, so Cameron in the lab applied this CRAC platform to study cell types in primary somatosensory whisker cortex. And he imaged animals during task, and he's focused on layer 2-3, which is the upper layer of cortex, looking at these different types of cells that were previously identified. And just to kind of skip to the chase here, what he identified was a cell type called BAS1A, which we believe operates as a circuit hub for sensory processing in this area. Now, one of the things that we noticed when we started to look through this data, and this kind of made us think, or at least made me think back in Ellie's time, was this idea of candidate plasticity genes. Genes are associated with neuroplasticity. what we found was when you look more closely at these BAS1A neurons, was that they were selectively enriched in certain sets of candidate plasticity genes, such as FOS or NPX2, which led us to wonder whether or not these particular types of cells have specific plasticity mechanisms that were specific to that cell type and others. So again, thinking back to my PhD days, one way to drive plasticity in these sensory systems is to perform deprivation experiments, such as individual cortex, you perform binocular deprivation. Since we're whiskers, we trimmed whiskers. So we perform these bilateral whisker deprivation experiments. We left a little bit of a stubble so we can still stimulate a whisker to see how sensory responsive the neurons were. And then we tracked two things. We tracked FOS expression across time, and we also tracked activity patterns across time and looked how they change within these different cell types. And what we found is, in general, if you look broadly in somatosensory cortex, when you deprive the whiskers to trim them, the activity patterns goes down, but there's a slight homeostatic recovery. If you look at the different cell types in S1, what we find is these BAS1A neurons, their expression of FOS doesn't track the activity changes. It remains stable across time. And these neurons themselves, when you perform the deprivation, they're the ones that show the specific homeostatic recovery, suggesting that potentially the genetic programs in this particular cell type somehow prime this cell type for plasticity in the face of changing experiences. So the idea is that this particular type of cell acts as a sensory hub in this area of cortex in order to maintain representations, but also to preserve these representations in the changes in the face of novel types of experiences. Okay, so hopefully that kind of gives you a clue into what we've been doing, but how really the past 16 years, and since I was at LA Labs, has really kind of somehow informed the things that we're doing. And it's exciting to think about what the next 20 years lies ahead, both in our lab, but also here at the Institute. And so with that, I'd like to just close by, again, acknowledging Ellie and all the people that have been in her lab for all the years, also people in my lab who've carried out the work, our collaborators and our funders. Thank you.
Well, Ellie, you trained him well. He's on time. Are there any questions from the audience to Jerry?
Okay.
Maybe I can ask one.
Okay,
since
sure.
I'm just taken aback by your timeliness. Have you tried to do spatial transcriptomics on the tissue after you've done the recording, as opposed to, piecemeal marker gene expression?
So you mean just by, that's kind of what we did, right? So after we imaged, we took the tissue out, and then we did the spatial transcriptive profile on the same tissue.
Right. I guess I meant like genome-wide transcriptomics.
Like really high read depth, you mean?
Yeah, looking at many genes.
Many genes.
Looking at 1,000 or 10,000
Okay,
genes.
yeah. So in our particular experiments, we're profiling on the order of 20 or 30 genes. And so we select those gene markers based off of the RNA sequencing study that kind of tells us, right, a priority, these are the genes that are defining the cells. But if we want to go a more forward approach where we kind of go more unbiased, we haven't yet ramped up to 1,000, but it's definitely on the table of things that we want to do. And I think it's possible.
Great. Okay.
Thank you.